Right. Thank you very much. We've got time for one or two questions, if there are any. Any?
Yeah, this is Andrew in Kampala, and I appreciate the talk by Dr. Marakalala. A very intriguing talk, and my background is infectious disease. I had a short question, and I think it's in the context where in Africa, a lot of TB occurs in the HIV context. And we also know HIV disease stage determines the way TB presents. For example, in severe forms, we see infiltrates usually in the lower part of the lung. I see your work has focused on granuloma formation, and I wonder whether you've had an opportunity to look at other disease pathogenetic forms like infiltrates in a setting of HIV disease. Thank you.
Sorry, just the last part of the question. That's what I missed a bit there. Sorry.
Yes, no, thank you. No, I noted that a lot of your work is on granuloma formation, but we know that with severe HIV disease, we tend to see infiltrates, usually in the bases of the lung. And I wonder whether you have looked at other forms that are not necessarily granuloma formation as part of your work, so that you can appreciate the impact of HIV on TB pathogenesis.
Okay. Yeah. So if I understood well, it's you want to see whether there are other forms other than the granuloma that's specific to TB that we've looked at that are associated with the disease.
Correct. Thank you.
Yeah. Okay. Yes. So the granulomas that we're looking at are mostly on TB, but of course, for control, for one to be sure that these mediators of disease are TB specific, one would need a control for other granulomas like sarcoidosis disease, or if there's actually cancer tissue, see that the staining of upregulation of these proteins are actually specific for TB response. And then, of course, it would also be important to look at to whether beyond granulomas that are whether there are other factors like as I showed in the blood in the different location, that you can also see those genes that would allow us to really develop biomarkers at point of care. So I hope I answered your question. Thank you.
Yes, that was helpful. Thank you.
Right. Thank you.
Any other questions? Well, if not, a few minutes, any questions for the earlier speakers this afternoon, bearing in mind that Aridazi is no longer here? No. Well, I think if there aren't, I think maybe the best thing, Mr. President, is if I now hand back to you, and then we can perhaps have the open-ended discussion under your chairmanship. But before that, could I thank most sincerely on behalf of all of us, the four speakers we've had this afternoon. I think we've been very lucky indeed to have such an excellent variety of speakers. So many thanks for all of them. Oh, just a minute. I think there may be a question from Salvador at this stage.
Yes,
Moncada, yes,
okay.
Yes.
For the last speaker, I wasn't very clear about how do you understand the arachidonic acid metabolites in granuloma formation. You talk about like oxygenase products, about COX-1 and COX-2,
yes,
which each of those pathways can be individually modulated. Do you know what is the effect of inhibiting, for example, COX-2 versus inhibiting the lipoxygenase pathway in granuloma formation, and in which way that affects the status of the granuloma in terms of infectivity, for example?
Yeah, that's a very nice question. Thank you so much. Yes, so what we know is that COXes, which lead to production of prostanoids like prostaglandin, thromboxane, as well prostacyclin, are largely, so while we know the pathway seems to be inflammatory, but compared to lipoxygenases that lead to leukotrienes and so on. So it's largely opposing the inflammatory leukotriene pathway. So the work that we have that came from genetics, but this is a mouse work, shows that when you knock out COX-2, you actually reduce disease severity. Indicate when you, yes. When you knock out COX-2 pathway, you actually make mice worse to the disease, indicating that COX-2 is important for protection against TB. But ALOX5 pathway, when you knock it out, you actually save mice from severity of the disease. So what we think, lipoxygenases are leading to too much inflammation that leads to damage in a granuloma progression, which can be intercepted when you actually promote the COX pathway that leads to production of prostaglandins that are associated with limiting the inflammatory mediators on the lipoxygenases. So our hypothesis is that while this pathway is also very, one has to be careful when you tamper with inflammation, because you also need some specific balanced amount of inflammatory mediators like TNF. It's only if it's too much and hanging around for too long that it leads to severe pathological damage. But when you actually don't have it at all, you also run in risk because it's also related to control of the pathogen. So I don't know if that makes sense.
Yeah, well, it's sort of, yeah. Thank you.
Thank you.
Well, and thank you, Dr. Marakalala. Thank you once again to all four speakers. We've had a good session, but let me now hand back to the President, who will chair the final open-ended discussion in our last half hour. Thank you very much again.